Hello everyone, myself Akshansh Mathur. I am from India. I am pursuing my master's in archaeology from Deccan College, Pune, India. My research area is to do research in the applications of archaeometry techniques like stable isotope analysis, lipid residue analysis, etc. techniques. Today I am talking about one of those technique. Name is stable isotope analysis. What is the use of this technique in the archaeology to solve archaeological questions? It is a photo of archaeological lab of Cornell Institute of Archaeology. The main aim of this study is to understand the uses of stable isotope analysis in the reconstruction of Paleodiet and what is its importance, what are the methods used under this technique to reconstruct Paleodiet, and what challenges this technique faces in today's time. It is the main part of the topic. It is a photo of an archaeological site. From these site, archaeologist gathers the ecofacts. Ecofacts, it means botanical environmental remains, which includes plant remains and animal remains. Before going down deep into the topic, it is very important to understand what is the basic meaning of atom and isotope. Atom is a very tiny particle of any non-living thing. All non-living things are made up of atom. Then atom made elements, molecules, and compounds. Atom contains three types of subatomic particles. In simple words, these subatomic particles made atom. These are electrons, protons, and neutrons. Protons and neutrons are present in the central part of an atom. That part is called nucleus. And electrons are revolving around it. This is the definition of atom. Means atom is a founding block of all non-living things. The second concept is what do you mean by isotopes? Isotopes are different kind of atoms of an same element, which contains same number of protons but different number of neutrons. As an example, carbon. Carbon is an element. It is made up of carbon atom. It is made up of three. It has three types of isotopes. These are C12, C13, and C14. All isotopes contain same number of protons, number six protons, but all isotopes contain different number of neutrons. Isotopes are of two types, stable isotopes and unstable isotopes. Stable isotopes cannot decay over time. On the other hand, unstable isotopes can decay over time. These are also known as radioactive isotopes. These isotopes are used in the techniques of dating, especially in radiometric dating. By studying the process of radioactive decay, archaeologists, archaeochemists measure the date of any artifact. Techniques like C14 dating method, potassium argon dating method, uranium series dating methods, all dating methods are rely on one concept, which is called radioactive decay process of any unstable isotopes like C14, like potassium argon, like uranium 238. On the other hand, stable isotopes are also very useful in the discipline of environmental archaeology with an aim to reconstruct past human ecology by using plant remains, animal remains, and human bone remains, reconstructing in the reconstruction of ancient substance pattern, etc. Stable isotope analysis is the implication of biochemistry. in the archaeology, which helps to give answers of archaeological and anthropological research questions. What type of stable isotope are used in archaeology to solve the mysteries of human evolution? Usually, today in this field, stable isotopes of carbon, oxygen, lead, strontium, nitrogen, sulfur isotopes are used to reconstruct following important archaeological information like paleodiet, like paleoclimate, like paleo-human ecology, ancient migratory pattern of animals and humans, along with the socio-economic context of past population. These type of information are reconstructed by using this technique. For the reconstruction of Paleodiet specifically, two types of isotope are in use. These are carbon isotopes and nitrogen isotopes. Carbon isotopes are very helpful to understand what kind of plants and food humans ate in their life in the past basically. And nitrogen isotopes are very useful to reconstruct what is the tropic position of past consumer. It includes both animals and humans. In the technique of stable isotope analysis, researchers usually use one concept. It is called delta concept. They use delta sign to express the value of ratio of two stable isotope in an sample. So it is very vital to understand what is the meaning of delta sign. Delta sign shows the relative difference of the ratio of stable isotope in an sample in relation to the standard material sample. As an example, here the word delta C13 is mentioned. It means the number of the ratio of stable isotope C12 and C13 in the sample in relation to the standard number. This is a photo of archaeological site where lab work are also done in that field to get instant result. The main research question for archaeologists to reconstruct paleodiet, as I said earlier, they uses carbon and nitrogen isotopes. If archaeologists found any bone from archaeological site, they can extract sample firstly from it like collagen, like minerals part. That from it they extract stable isotopic data of carbon and nitrogen isotopes. They try to measure the ratio of C12 and C13. Then they measure and find the value of delta C13 by using following mentioned formula. If the value of delta C13 is higher but more negative, higher means values if value is higher and less negative, it means that human whose bone archaeologists find ate more C4 plants than C3 plants. Because C4 plants are always enriched in C13. That's why their value is less negative and higher. Their delta C13 value may be vary from minus 21 parts per million to minus 26 parts per mil. But if sample shows value is very low but more negative, it means human population ate more C3 plants than C4 plants because C3 plants are more enriched in C12 than C13. That's how by using carbon isotopes, archaeologists reconstruct paleodietary pattern. What is the importance of this method? This method is very helpful to reconstruct paleodietary pattern. This method is very useful to reconstruct paleodietary pattern by using stable oxygen isotope. For reconstruction of paleodiet, O16 and O18, these two oxygen element stable isotope are used. If delta O18 value is higher, it means dry climate. If value is low, it means wet climate. That's how this technique is very helpful to reconstruct paleoclimate or paleoenvironment. This method is very useful to understand the waning age. This concept helps archaeologists to understand socio-economic aspect of ancient people. This method tries to distinguish the importance of marine diet and terrestrial food diet in the ancient life of humans, because marine organisms or marine plants are always enriched in C13, which is heavier isotopes, because they take carbon directly from dissolved carbonate from ocean, or dissolved carbonate is always enriched in C13. That's why marine plants are always enriched in C13. That's why they always show higher value and less negative delta C13 value. On the other hand, marine plants are also high and enriched in N15, because they take nitrogen directly from dissolved ammonium nitrate. So this technique is very helpful to distinguish the importance of marine diet and terrestrial diet of past people. This approach gives quantified and scientific data to understand the human interaction with animals and plants. It is the importance of this method. Various samples are used in archaeology to extract stable isotopic data. As we know, human bones are made up of two specific parts. One is called organic part, other is called inorganic part. Organic part contains collagen and non-collagen. Inorganic part contains minerals, calcium, phosphate, all of these things. And hydroxyapatite, it also mineral found in the human bones. Collagen minerals are used as a sample for the extraction of stable isotopic data. Various methods are used to extract collagen, especially these are Seeley method, Torus method, and Longin method. Longin method is a very useful method to extract collagen from bone. Under this method, bone are dipped in the firstly, our scientists put bone in the strong acid called HCl. HCl dissolve hard mineral part, and only soft part, which is collagen, left. After that, archaeologists put that soft collagen part into a machine called elemental analyzer. This machine converts solid sample of collagen into liquid sample. Then that liquid sample put into the new machine called IRMS. It is IRMS, which is used to measure the ratio of C12 and C13 isotopes in a sample. It has four components: inlet system, source, analyzer, and collector. This machine helps archaeologists to measure the ratio, ratio of two stable isotopes, which is C12, C13, and 14 and 15 in an sample. What are the challenges this technology face in today's time? The first biggest challenge is preservation of sample in an archaeological site within the geological context. Due to physical reasons, chemical reactions, environmental reasons, human factors always affect the sample preservation in an archaeological site. Second, at various times, archaeologists found very small fertile bone. If you want to extract collagen or many other part of minerals from the small fertile bone, it destroyed them. So it is very difficult to extract sample from the bone. This is the second biggest problem that archaeologists face in today's time. Third problem is poor preservation of samples in the lab due to time issue, money issue, and skill issue. Fourth problem is regional variation in the baseline of isotopic value because due to climatic variation, environmental variation, every region has its own isotopic value. These are some problems and challenges which archaeologists face by using this technique. Hence, this technique proves very useful in archaeology to solve various archaeological questions, to solve the mysteries of human evolution. Despite it contains various challenges, its future is very great and well because this technique is very vital to solve the problems which arises in the front of archaeologists to understand how climate affect the human and how human affect the climate. To understand the relationship between humans and animals, humans and environment basically, this technology proves very, very useful. Thank you very much.